Good afternoon, children.
Good afternoon.
We were discussing about the transcription process taking place in prokaryotes in the previous class, and I told you there are three major steps involved. What are the three major steps, Sania? Three major steps in transcription.
Initiation, elongation, and termination.
Yes. And name the enzyme involved in the process. Gayatri, which is the enzyme involved in the process?
RNA polymerase.
Correctly. RNA
polymerase.
Polymerase. What adjective you can add?
DNA dependent.
DNA dependent RNA polymerase is the enzyme catalyzing the reaction. What is the significance of termination factor and initiation factor? Anna, what role do they play?
Ma'am, last class I was not there.
Nice excuse. You were present or you were absent?
Absent.
But I think you joined late. No. What happened?
I was not feeling well. Stomach was upset.
Okay. Steffi, can you tell me what is the significance of these factors?
In initiation, RNA polymerase binds to the specific site in the DNA called the promoter, and in termination, the RNA polymerase they will recognize when it is about to end.
Okay, that means RNA polymerase, they can catalyze only elongation process. If they have to recognize the promoter and the terminator, they need the assistance of these factors. Only in the presence of these factors, they can recognize the promoter gene and start the process, recognize the termination gene and end the process. Okay, so shall we proceed? Yes. In prokaryotes, transcription and translation. Transcription and translation. Translation we haven't learned. We are going to learn about it. But transcription, you know. Transcription means, Anna, what is transcription? This we learned last class, not today. Simple sentence, single line answer I want. What is transcription?
Process of copying of genetic material from one DNA to other. One strand.
DNA to? Louder.
One strand of DNA into RNA.
Good. So you are remembering. So it is a process by which genetic information is copied from DNA to RNA. And what is translation? Translation we haven't learned. It is a process by which the RNA is forming proteins, or it is translated into proteins, and transcription process will be followed by translation. In transcription, RNA is formed, and in translation, that RNA will get converted to proteins. So transcription process will be followed by translation, and both of them will be taking place in the same compartment, because there is no difference between cytosol. Cytosol is same as cytoplasm. The cytoplasm in prokaryotes, so you call it as cytosol. Why there is no difference between cytosol and nucleus? Why this is mentioned, that there is no clear demarcation between cytosol and nucleus in prokaryotes? What is the reason? Simple. Think about the definition for prokaryotes. You will get the answer. Why your videos are off, children? Don't want attendance? Anthony, Nia, Evelyn, where Vidur went? Yes. Give me the answer. Think and say why it is said that there is no demarcation, there is no boundary or no separation between cytoplasm and nucleus. What is actually separating cytoplasm and nucleus? What separates nucleus? Normally in a cell, how are they separated? If that is not there, both will get mixed up. Both will have no boundary or separation. What is that? What is that membrane?
Cell membrane.
Louder. Cell membrane is the outermost boundary. In between cytoplasm and nucleus, which is the membrane? Naureen, which is the membrane?
Nuclear membrane.
Nuclear membrane. Vidar. Nuclear membrane is separating nucleus and cytoplasm. But we already learned that in prokaryotes, there is no Vidar. Vidur is sleeping in between. He's not there. Nandana,
Yes, ma'am.
you just explain it why it is said so. There is no difference between cytosol and nucleus.
Because the nuclear membrane is not present.
Not present. So both will, we cannot separate out the cytoplasm and nucleus there because they have some genetic material and that genetic material is not having a boundary or nuclear membrane. Okay. And hence both these processes will be taking place in the same compartment. Whereas in eukaryotes, there is difference. Translation is taking place in nucleus. Transcription is taking place in the translation is taking place in the cytoplasm and transcription is taking place in the nucleus. But here in the case of prokaryotes, there is no such difference. Both are taking place in the same compartment. Therefore, translation can begin even before transcription is completed. So in the case of prokaryotes, they don't have to wait for the transcription process to get completed because both can take place side by side. Even before the completion, even before the messenger RNA or even before the RNA is formed, they can start the process of translation because the site is same. These are taking place. These two important processes are taking place in the same compartment because there is no nuclear membrane to separate cytoplasm from nucleus. Whereas in the case of eukaryotes, the situations are different. There we have a well-defined nuclear membrane which will separate nucleus from cytoplasm. Are you getting it? Are you following? Children, I have told earlier also, when I ask you something, please respond. Can you follow?
Yes, ma'am.
Thus, in prokaryotes, transcription and translation are coupled, means it takes place together. All these points as application level questions it can be asked. Why transcription and translation processes are coupled in the case of prokaryotes, means both are taking place together. Normally, transcription should be completed, only then translation can start. But in prokaryotes, both can take place together. It's not necessary for the transcription process to be completed for translation to start. That is because they don't have a nuclear membrane to separate cytosol and nucleus. Hence, both processes are taking place in the same compartment. But in the case of eukaryotes, the case is different. Their translation and transcription are taking place at different sites. Translation in the cytoplasm, transcription process in the nucleus, because we have or eukaryotes are having a well-defined nuclear membrane. Hope you have understood. Any doubts? Let me know. Eukaryotes. 3 different kinds of RNA polymerases are present. How many types of polymerases we discussed in the case of prokaryotes, children? How many types are there in prokaryotes? Rijas, how many types of RNA polymerase we discussed in the case of prokaryotes?
3.
3. Prokaryotes.
3.
3. Which are they? You are referring the textbook, no? That's why. Sania, how many?
3. Ribosomal, messenger, and trans.
Did you hear my question? RNA polymerase. I didn't ask about the RNA enzyme. How many types of RNA polymerases did we discuss under prokaryotes? One, just one. Hope you are remembering that sentence. I'll show you that slide. If you're forgetting what we learned in last class, and once the chapter is completed, what will happen, children? Polymerases are present. RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. Whereas in prokaryotes, there is just one single DNA dependent RNA polymerase. But in eukaryotes, in higher organism, 3 types are there. They are RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. And each one is having a specific function. RNA polymerase 1, it will transcribe ribosomal RNA. rRNA means ribosomal RNA. Then RNA polymerase 2, they will transcribe hnRNA. hnRNA means heterogeneous nuclear RNA. That is actually the precursor of messenger RNA. Once transcription is completed, once transcription process is completed, what we are getting is hnRNA. Later, this hnRNA undergoes different processes and then it will change into messenger RNA. That is why it is mentioned. This is the precursor of messenger RNA. When hnRNA undergoes certain transformation, it will change into messenger RNA. So which enzyme is concerned with the transcription of hnRNA? RNA polymerase 2. Then RNA polymerase 3. It's concerned with the transcription of transfer RNA, 5S ribosomal RNA, small nuclear RNAs. So it can be asked which are the different types of enzymes present in eukaryotes, give its function, or which are the different enzymes that carries out transcription process in eukaryotes. So you should be familiar with all these. Once again, in prokaryotes, there is just one RNA polymerase enzyme, but when it comes to eukaryotes, 3 types are there. RNA polymerase 1, 2, and 3. Each one is having a specific function. RNA polymerase 1 will bring about the transcription of ribosomal RNA, whereas RNA polymerase 2 will carry out the transcription of hnRNA. hnRNA. What is its significance? Steffi, what is the significance of hnRNA?
It is the precursor for
precursor of messenger RNA, which means first when it is formed, it will be in the form of hnRNA. Then it will undergo some transformation process and will change into messenger RNA. And then RNA polymerase 3, it's concerned with the transcription of transfer RNA, 5S ribosomal RNA, and small nuclear RNAs. Details of that you don't have to learn, but you should know what RNAs they are transcribing or what is its role. Each of these enzymes role you should be thorough with. Okay? Yes? Shall I change?
Yes, sir.
Okay. Complexities associated with transcription. Now let's see what are the different complexities which are associated with the process of transcription. In eukaryotes, monocistronic structural gene. One of you tell me what is meant by monocistronic structural gene. We have already learned. Vidar, you came back. Children, I will not continue if your videos are off like this. Vidhur, tell me what is monocystronic gene. We have learned about monocystronic and polycystronic genes. You can refer and tell me. Find out. At least you should know where it is.
The eukaryotic structural gene with polypeptide or protein.
Which will code for?
Polypeptide.
How many polypeptide? That makes a difference.
One.
So monocystronic genes are those which code for just one polypeptide or protein. When it is coding for many polypeptides, what do you call it? You're not well, Vidhur? Not feeling well?
No.
Only hearing my voice, you became sick? Okay, Punya, what is polycystronic gene?
Many polypeptides are coded by it.
Not audible. Once again.
Many polypeptides are.
It will code for many polypeptides. So in eukaryotes, the monocystronic structural genes, they have interrupted coding sequences and you call the genes as split genes. So in eukaryotes, some interrupted, the coding sequences are interrupted. Some unwanted sequences are there in between and you call these genes as split genes. So in that structural gene, in monocystronic structural gene, the entire gene is not required. You call them as split genes because some unwanted sequences are also present in between. The coding sequences or expressed sequences you call as exons. Exons are interrupted by introns. That means monocystronic structural gene consists of exons and introns. Exons and introns are present in these genes and exons are needed and introns are not required. Okay, in that particular gene, coding sequences or those sequences which have to be expressed, they are present. Along with that, something which need to be removed are also present. Some interruptive sequences are also present. So what is to be done? Those should be removed off. Introns should be separated and exons should be joined together. Children, see the figure over here. Just a minute. Okay. See, this is a RNA. And here you can see this is exon, in between intron, then again exon. And these sequences are unwanted sequences. So what is to be done? See, what is shown in blue color are only required. What is seen in yellow color are not required. They are interruptive sequences, not required. So in the second step, you can see that is being removed off and only exons are being joined together. So the unwanted sequences are removed and the required sequences are joined together. I'll explain it again. In eukaryotes, the monocystronic structural genes, they have two types of sequences, coding sequences and some interruptive sequences. And hence we call them as split genes. It's not continuous. In between some unwanted or interruptive sequences are there. So you call them as split genes. And split genes consist of exons and introns. The coding sequences or expressed sequences you call as exons. And in between the exons or interrupting or disturbing the exons, there are certain other sequences you call them as introns. Exons are those which will appear in a processed messenger RNA or mature messenger RNA. Children, when I showed you the last slide, I told hnRNA is the precursor of messenger RNA. Okay, and that hnRNA will undergo several modifications and will transform into messenger RNA. And exons are those sequences which you can find in that mature RNA. Those which are appearing in the mature RNA are exons. So then what will happen to introns? They will be cut off. They will be removed off. Introns, easy to remember, intervening sequences or interruptive sequences. They never appear in mature or processed messenger RNA. They'll be spliced out. They'll be removed off. They'll be cut off because it's of no use. It's not required. It's not having any specific function in this RNA, so it will be spliced off. So that is known as splicing. The primary transcript. Primary transcript means as soon as transcription is over. So we learned about the transcription process. So there I told you, as soon as the transcription process is completed, the messenger RNA, the RNA and the enzyme will get separated off. And that RNA which is formed immediately after transcription, you call it as primary transcript. It's also having one more name. Already we discussed hnRNA or heterogeneous nuclear RNA. So in that RNA, both exons and introns will be present. And later it will undergo, I told you several transformation or some changes it will undergo. Only then this precursor of messenger RNA, that is hnRNA, will change into messenger RNA. Clear? So in hnRNA, both exons and introns will be there. But once it is polished or once the processes are completed, we get messenger RNA. And in messenger RNA, only exons will be present. Introns will be removed off. What do you call the process of removing them? Splicing. So the primary transcript or the messenger RNA, hnRNA will be having both exons and introns, but later it will undergo a process known as splicing, by means of which that interruptive sequences or introns will be removed and exons will be joined together. You want me to explain again? Shall I explain? Okay. So we learned about transcription process. So once transcription is completed, I told we get an RNA. An RNA strand will be formed. But this RNA formed immediately after transcription, you call it as primary transcript. Formed as a result of transcription. Primary transcript or hnRNA, heterogeneous nuclear RNA. So that will undergo certain transformation, some changes it will undergo before it becomes the matured RNA. This much is clear? Yes. Now, what is special about, or what is the main difference between hnRNA and messenger RNA? In hnRNA, along with the coding sequences or along with the sequences that will get expressed, some unwanted sequences are also there. The expressed sequences, you call them as exons. And those sequences which are interrupting, you call it as introns. So in hnRNA, exons will be there, along with that introns also will be there. But we don't need introns, we need only exons. So what has to be done? Those introns should be removed. See the figure? The introns should be removed and the exons should be joined together. See, exons, intron. This intron is removed off and then the exons are joined together and then only we get the messenger RNA. When both are present, immediately after transcription, you don't call it as a messenger RNA, you call it as hnRNA, which will undergo different procedures to get transformed into messenger RNA. More procedures are there, you're going to learn about it. Is this clear? Difference between exons and introns? Which one is needed, introns or exons?
Exons.
Exons are needed. What is the difference between hnRNA and messenger RNA? What main difference, Anna? How are they different?
Coding sequence.
What is the other name for coding sequences here?
Exons.
Exons. Interruptive sequences? Introns. And which one should be removed? Steffi, which one should be removed? Exons or introns?
Introns.
Introns should be removed and exons should be joined together. And Anthony, Donna, tell me what is the process by which we remove these introns? What do you call the process?
Splicing.
I'm not able to hear you. How do we remove? What is the procedure or the method by means of which exons are joined and introns are removed?
Splicing.
Splicing. You're not audible, but from your lips I could make out that you said splicing. Splicing is the process. So for 1 mark and all, if it is asked, you should know. Application level questions can come. You know, objective type questions are there. You should be so thorough with the textbook. Each and every line. There is not even a single line you can say is less important and avoid. Everything you have to read. Okay, each and every line, including the footnotes of the diagram that I have told you from the beginning. Okay, we'll continue. Introns are removed and exons are joined by a process called splicing. The name of the process is splicing. What is splicing? It's a process by means of which introns are removed and exons are joined together. And exons and introns both will be present in hnRNA, but once it becomes matured RNA, that is, hnRNA will undergo splicing and then it is getting transformed into messenger RNA. So messenger RNA will not have introns. It will have only exons. Clear? You want me to repeat again? Is that fine, Evelyn? Okay. Now, 2 more processes are there which the hnRNA has to undergo. Not only splicing, it has to undergo some other process also before it can become the matured messenger RNA. This is also very important, children, very important topic. One is capping and the other process is known as tailing. Capping and tailing. 2 more procedures apart from splicing. hnRNA, heterogeneous nuclear RNA, should undergo splicing. Definitely we learned about it. Other than that, capping and tailing. Only then it can become mature RNA. Now let's see what is meant by capping. In this, an unusual nucleotide, the name of the nucleotide is methyl guanosine triphosphate. It will be added to the 5' end. So 5' is considered as the head and 3' as the tail. So when you add something at the 5' end, you call it as capping. And when something is added at the 3' end, it is tailing. So in capping, methyl guanosine triphosphate. Methyl guanosine triphosphate. Where it is added? To the 5' end of hnRNA. You should not write at the 5' end of messenger RNA. hnRNA. Only when splicing, capping and tailing procedures are over, it will change into messenger RNA. So first step we learned splicing, removing introns, joining exons. Second step, capping. Hearing the term cap, 5' end. You know that 5' end polymerization is also taking place from 5' to 3' end. Assume that 5' is the cap or the head and the 3' end is the tail. So at the 5' end, when methyl guanosine triphosphate is added, you call it as capping. Similarly, at the tail region also, you add adenylate residues. Residues of adenylate around 200 to 300 will be added at the 3' end. In a template independent manner, not in any particular order, these will be added at the tail end or the 3' end. So in capping, at the 5' end. In tailing, the change is made at the 3' end. And in capping, what we are adding? Methyl guanosine triphosphate. And in tailing, we are adding adenylate residues. How many? Hundreds of them are being added. Children, when you are preparing a magazine or when a book is being, prepared or made, after pinning together all the pages where the matter is there, you will add a cover page and a back page. Am I right? So just like that, once the process is completed, to give it additional protection, we include a cover page and a back page. So same is the case here. The main purpose is to provide them protection. So the 5' end will provide protection from degradation, and it also helps the translation process. Whereas the tail end the tailing process also will protect it from degradation, and it also helps in transporting the messenger RNA into the cytoplasm. I told that the transcription process is taking place in the nucleus. From there, it should be transported to the cytoplasm. So the tailing process also helps in transporting this messenger RNA to the cytoplasm, plays an important role in translation. Main purpose is to protect it from degradation, protect it from getting damaged or degraded. Details you don't have to learn. So both capping and tailing will give them additional protection from degradation. So once again, once the transcription process is completed, we get primary transcript. What is the other name for primary transcript, Meenakshi? Primary transcript is also known as which RNA? Relevant? Which RNA? Michael, turn your video on. Yes, Meenakshi. Meenakshi is crying hearing my question. Which RNA? You're not well? Huh? Only when question is asked or you have cold? Common cold is there? Okay. But I did not hear. You speak a little louder. HNRNA. What is the expansion of HNRNA, Naureen?
Heterogeneous nuclear RNA.
Heterogeneous nuclear RNA. Okay, heterogeneous nuclear RNA. Michael, can you tell me what are the three processes this HNRNA has to undergo before becoming messenger RNA? Michael? Audio is working now. Yes.
Splicing.
Yes.
Capping.
Very good.
Tailing.
Tailing. You are not reading, no? Splicing, capping, and tailing. And what is splicing? Mehek, what is splicing?
The introns are removed
and
are joined.
Yes, good. Then what is meant by capping, Lydia? Capping?
Methyl guanine is added to
methyl guanosine triphosphates are
added to 5' end of
that is known as capping. Then what is tailing, Anthony?
Adenylate residues.
Adenylate residues. Where are they added?
3' end of
to the 3' end. How many residues are added?
200 to 300.
200 to 300. Good. So we can continue, no doubts. Can we continue? Okay. When HNRNA is fully processed, now three major steps they have undergone. In many question papers, I have seen these three steps. Since yours is objective questions, you will not get such a question now, but as one word question, definitely you can get. Usually for 3 marks, they used to ask what are the different processes by means of which HNRNA is getting converted to messenger RNA. So once these steps, these 3 steps, splicing, capping, tailing are completed, HNRNA will be processed, fully processed, and it will form what you call as messenger RNA. Now this can be transported from the nucleus to the cytoplasm. Where is transcription taking place, children? In eukaryotes? Steffi, where is transcription taking place? Transcription, DNA. Genetic information copied to RNA. Where is DNA present?
Inside the nucleus.
Nucleus. And translation is happening in the cytoplasm. So once messenger RNA has been formed, it should be transported to cytoplasm. From the nucleus, it should be transported to cytoplasm so that they can take part in the translation process. The split gene arrangements represent probably an ancient feature of genome. The split gene arrangement. What is meant by split gene arrangement? The presence of introns and exons. Both are present. Even though introns are not needed, their presence is also noted. So that split gene arrangement represent an ancient feature of genome. The presence of introns, same sentence from your textbook. The presence of introns is reminiscent of antiquity, and the process of splicing represents the dominance of RNA world. The presence of introns is reminiscent of antiquity, something which existed in the past. Reflections or something which was left over from the past, that is indicating, or that is indicated by the presence of introns, which means that for the formation of RNA, or till the formation of this RNA, it was required. So it might have had some function earlier, but once the RNA has been formed, it is no longer functional. So they might have some function in the past. And the process of splicing represents the dominance of the RNA world, because RNA decides whether introns should be present or not. So that is why it is said it will indicate the dominance of RNA world. It decides that introns should be removed and exons should be joined together. So this sentence reminds us about the reminiscent of antiquity, means these introns might have had some significant function earlier, but once the RNA has been formed, it is of no use. And the dominance of RNA world is clear from the fact that RNA is deciding whether this intron should be present or not, and it is undergoing the splicing process to remove these introns. Any doubt in that statement?
Ma'am, could you explain it once?
Okay. See the split gene arrangement. Split gene arrangement means the genes are split. How the genes are split? Exons are split or separated by means of introns. And you know the coding sequences are only exons. And the unwanted or interruptive sequences are introns. And these introns, the presence of introns indicate that, or the split gene arrangement indicates some ancient feature, some old feature of the genome. So these introns might have had some function earlier, but after the formation, once this RNA has been formed, it is of no use. So that presence indicates that they had some function in the past, or it indicates some memories of the past or some reflection of the past. It had some significant function earlier, but it is no longer functional. And you know that before the RNA is getting formed, hnRNA will undergo the splicing process by means of which introns are removed and exons are joined. So the RNA is deciding whether the introns should be maintained or not, and that is getting removed off. So that indicates, since RNA is deciding the splicing process, we say that this indicates, this process is indicating the dominance of the RNA world. Okay? You read it, and still if you have doubts, next class we can discuss again. Okay? You read the textbook. Any doubts, let me know. Shall I wind up?
Okay, ma'am.
Any doubts? Any doubts? Splicing, capping, tailing? Punya will come to that. Next class we can discuss. Is there any doubt? Splicing, capping, tailing? No doubts. Clear. What is hnRNA? Different types of RNA polymerases. Which are the 3 types of RNA polymerases, Mehek? Names, only names. RNA polymerase.
Ma'am, can you repeat the question?
What are the different RNA polymerases in eukaryotes? Just number them.
RNA polymerase 1, 2, and 3.
RNA polymerase 1, 2, and 3. Which enzyme is playing an important role in the transcription of hnRNA?
RNA polymerase 2.
Yes, RNA polymerase 2. We can continue next class. Any doubts, do let me know. Okay. Nia is present then. Okay, thank you children. See you next class.